right, so here we are in chapter 8.5, and we are talking about the catabolism of lipids and proteins. So far in Chapter 5, what we've spoken a lot about is the catabolism of glucose, or utilization of carbohydrates, namely glucose, in being broken down for energy. So we've talked about glycolysis, the Krebs cycle, the ETS. We've talked about aerobic respiration, anaerobic respiration, fermentation, all of these different things. But it's not only glucose or carbohydrates that are broken down for energy. Just like in humans, we can break down other things for energy as well, and so can bacteria, or so can other microorganisms. So we're going to talk about the breaking down of lipids and the breaking down of proteins. So all of the steps to the process of breaking down these things, and then how these can be utilized for energy. So this is similar if you've had any experience in previous courses related to humans and how we break down lipids and proteins. We see that microorganisms do things that are similar in breaking down the lipids and proteins, and then what we're doing in breaking these things down is we're connecting to the carbohydrate catabolism. So we're going to take these products of lipid breaking down and protein breaking down and put them into things like glycolysis, or the transition reaction, or the Krebs cycle, or into the ETS, or oxidative phosphorylation, that kind of whole system at the end there, and then utilize them for energy. So let's first take a look at lipids. So when we take a look at lipids, we can talk about triglycerides and phospholipids. Remember, however, that lipids, when we talk about triglycerides, specifically what we're talking about is long-term energy storage. So in humans, for example, we store triglycerides in our body in adipose tissue, and that's for our long-term energy storage. So once we've gotten through our carbohydrates, our stored carbohydrates in the form of glycogen, then we move on to our triglycerides, our long-term energy storage. And microorganisms can do the same thing. They can utilize triglycerides for long-term energy storage. They can also break down the triglycerides of other organisms, and we'll see that in just a moment. So triglycerides, and remember that a triglyceride, tri meaning three fatty acids on a glycerol backbone. So if we remember just kind of my general drawing here, this would be our glycerol backbone, and then we have our three fatty acids coming off of this. I just draw our general carbon structure, and then I put R, meaning that continues. So our glycerol backbone are three fatty acids. So these are our fatty acids that are attached to that glycerol backbone. The other type of lipid that we're going to talk about are phospholipids. And of course, phospholipids are part of our membrane, plasma membrane, as well as plasma membrane for microorganisms. So they utilize phospholipids for their plasma membrane. So they can utilize phospholipids from their plasma membrane for energy. However, when we are talking about breaking down phospholipids, or the catabolism of phospholipids, it's mostly related to production of enzymes to break down other organisms' phospholipids. So getting inside of other organisms, or bursting the cell of other organisms by utilizing enzymes to break down other organisms' phospholipids, or other cells. So when we take a look at phospholipids, if we remember, there's a phosphate group, and then that's attached to a glycerol backbone. And then in this case, we have two fatty acids. So not three, that's the triglyceride. We have two fatty acids here. So our phosphate group, our fatty acids, and then our glycerol backbone here. So when we are talking about catabolizing these things, again, remember that these types of metabolic pathways, I should write this up here, these metabolic pathways are porous. What that means is it's not a closed loop. So substances can enter and they can leave for different pathways, or for other pathways. So we can take things, and I mentioned this already, a lot of these products are going to go right into either glycolysis, the transition reaction, Krebs cycle, ETS. So we can say what this means is that substances enter and leave for other pathways. And we'll take a look at that right now. So our triglycerides, what we are going to see is we are going to utilize what are called lipases. So ACE is an enzyme. So we have our lipids that are broken down by lipases. ACE meaning enzyme, the enzyme that breaks apart lipids. So our lipases are going to break off, they're going to cleave off these fatty acids here. So then what we end up with is the glycerol backbone and 3 fatty acids. So that's what we start with the lipases. And so when we're talking about phospholipids, we see something similar happen, but in this case, they're actually called phospholipases. Again, enzyme, ACE. And then they are going to not only cleave off these 2 fatty acids, but we're also going to cleave off that phosphate group. So what we get at the end here is we get our glycerol backbone, we get the phosphate group, and then we also get the 2 fatty acids. So this is the first part of lipid catabolism. So we've gone in, we've cut off those fatty acids, and in the case of the phospholipids, we've cut off the phosphate group. So then the bacteria is going to move on further from that, and it's going to continue breaking things down. So for example, with the glycerol backbone, what it's going to use is it's going to go in, it's going to change it. So we're going to say change, and it'll eventually change it to glyceraldehyde 3-phosphate, or G3P. So it's going to turn that glycerol backbone into glyceraldehyde 3-phosphate, and if you remember where we see glyceraldehyde 3-phosphate or G3P is in glycolysis. So remember, the G3P is the end product of the first portion of glycolysis, the energy investment stage. The G3P then is going to go through the energy harvesting phase to get to pyruvate at the end, and then we end up getting those 4 ATP, but netting 2 ATP. In this case, since we're skipping the investment piece, we actually are netting 4 ATP from this glycerol backbone because we're going in at the glyceraldehyde 3-phosphate in glycolysis. So we can see that down here as well. So from our phospholipases breaking off that glycerol is going to be converted into G3P, and it's going to go into glycolysis and net us 4 ATP. So then we have these fatty acids. So the phosphate group is going to kind of go off and do its thing, be utilized for phosphate and oxygen and things like that, or another phosphate group. So if we're going to be building phospholipids in this organism, then it can use that phosphate group to build new phospholipids for its own plasma membrane, for example. So it'll recycle that phosphate group. What we then have are the fatty acids. So the fatty acids go through a process called beta oxidation. And in beta oxidation, what it does is it removes the acetyl groups from those fatty acids. And then in that process of removing the acetyl groups, we actually reduce NAD plus and FAD. So let's see, so process of removing the acetyl groups. So we have our acetyl, that's the fatty acid part. Acid is the acetyl group there. So it's going to remove the acetyl group, and then the acetyl group is going to go with coenzyme A. How about I move this over? So the acetyl group is going to go with coenzyme A, and then it's going to go into the transition, or is part of the transition reaction that is going into the Krebs cycle. So this coenzyme A with the acetyl group is the transition reaction. So maybe that's not the best way to put that. So this is the transition reaction, and then it goes into the Krebs cycle. And so then when it goes into the Krebs cycle, of course, what we're doing is we're getting more ATP, we are getting more NADH, and we're getting more FADH2s. So all of those things are going to produce more energy for the organism. Now, when we remove these acetyl groups over here via beta oxidation, in that process, we're actually taking NAD plus and converting it into NADH. And then we're also converting FAD into FADH2. So all of these things can go into the ETS. So the ETS over here, these also can go into the ETS, or oxidative phosphorylation, however we want to say that. And then get us more energy, or get this organism more energy. So that's how we break down our lipids, and then we see the same thing down here with the phospholipases, right? So we saw the glycerol backbone turn to G3P, went into glycolysis to make 4-ATP. This phosphate group can be recycled, and then we have our 2 fatty acids. So again, they go through beta oxidation. We take our acetyl group, and those acetyl groups, there's 2 of them because there are 2. I should probably put that there too. So here we have 3 acetyl groups because of the triglyceride, because of the 3 fatty acids. Here we have 2 fatty acids, so we have 2 acetyl groups, and those 2 acetyl groups are going to be converting our NAD into NADH. Also, in this process, we will be reducing FAD to FADH2. And then in this process with our CoA, then we have acetyl-CoA that goes into the Krebs cycle. Just like what we saw, exactly what we saw up here with the triglyceride. So then these things are going to go to the ETS, or oxidative phosphorylation. Over here in the Krebs cycle, we're making ATP, NADH, and FADH2, which then are going to go to the ETS, or oxidative phosphorylation. So you can see why, just by taking a closer look at this process of lipid catabolism, why lipids are considered our long-term storage, our long-term energy storage, because we can go in with our original lipases. So lipases and phospholipases can cleave off those fatty acids. We can use the glycerol backbone to go into glycolysis. We can use those fatty acids then to go in, and when we break apart those fatty acids, then we get that acetyl group that's going to get us more NADH and FADH2 and ATP, and then the rest of it, or as during that transition into the acetyl groups there, beta oxidation, we actually produce NADH and FADH2. So that's going to go into the electron transport system, or oxidative phosphorylation. So we're really getting a lot of energy from these fatty acids here. And even the glycerol backbone here gives us some energy. So you can see why lipids are such a high energy molecule. So not only are they a high energy molecule, meaning good for the organism that's doing that, particularly if we're talking about bacteria, for example, they're getting lots and lots of energy by breaking down triglycerides, breaking down phospholipids, but these lipases and phospholipases are going to add to the virulence of the microorganisms. So they add to the virulence of the microorganism. And this is only for the microorganism that has these types of enzymes, the lipases and phospholipases. So that not only is one way that we can determine what organism we have, if we already know a particular organism does or does not have a lipase or phospholipase, but if it does, it adds to the virulence because, as I've kind of been mentioning, it allows them the ability to catabolize something other than carbohydrates. So one, it can catabolize something other than carbs. So it doesn't have to rely on something like glucose or lactose. It can switch and do something like triglycerides or phospholipids. But two, it can get into cells. So get into cells more easily. Because if it can just utilize its lipases and start to break down those phospholipids on the plasma membrane, then it can get itself right into the cell. So it's very, very good for a microorganism to have these types of things. Good for the microorganism, perhaps not good for something that it might infect, like us as humans. So now let's take a look at protein catabolism. So when we're taking a look at proteins, recall that proteins are chains of amino acids at its core. So we have the primary, secondary, tertiary, and quaternary structures of proteins, and recall that the primary structure is just a chain of amino acids. And all of these amino acids have an amino group on them, which gives them their name. And then, of course, we have all of these reactions to fold them up at various levels, which are the secondary, tertiary, and quaternary is joining it with other chains of protein or chains of amino acids. So we're going to focus on the actual chains of amino acids, which is the primary structure of proteins. And the way that organisms are going to catabolize these things are by utilizing proteases. So again, ACE and then protein. So enzymes that break down proteins. So first we can talk about extracellular proteases. So extra meaning outside of, cellular. So enzymes that are outside of the cell that can cut proteins internally at specific amino acid sequences. What this does is that it's going to produce chunks or fragments of peptides that can then be taken up by the cell. So enzyme outside of the cell that can cut proteins. Of course, more specifically, what we're talking about are cutting these amino acid chains internally at specific sequences of amino acids, producing fragments or peptides, which are small chunks of amino acid chains that can be taken up by a cell. So these bacteria, they're microorganisms that have external or extracellular proteases, have enzymes that are hanging out on the outside of their cell. So basically, again, this is a situation where they can burrow through proteins. They can break apart the proteins in order to get in somewhere. So an enzyme that's outside of the cell that can cut apart these proteins and then get at something or get in somewhere. But then also can utilize these pieces and these chunks, which we'll see in just a moment, for energy. So twofold, kind of in the way that we saw lipids. One, we can get energy, or the organism can get energy from these proteins, breaking them down. But then secondarily, they can get into places, so it increases the virulence as well. And so similar to the lipases that we spoke about, or the phospholipases, or even some of the enzymes that we talked about with glycolysis, certain bacteria, certain microorganisms have certain extracellular proteases and some do not. And so we can identify organisms, again, based on the different types of proteases, the different things that they can break down. So for example, some pathogens produce specific proteases. Whoops. And then we can ID based on this. So for example, gelatinase or gelatinase, it is produced by Proteus and Serratia genera. And so what we can do is we can see about determining what type of genus it is, kind of at least separating out by genus, even if we can't get to the species yet. But we can see if it fits in the genus Proteus or Serratia if it breaks down gelatin. So gelatinase, ACE meaning an enzyme, and gelatin. So if it breaks down gelatin, so that it won't solidify. So we can have gelatin, and if we add the bacteria to the gelatin, if we use an inoculating needle and we push the bacteria within the gelatin and then incubate it for 48 hours or so, then if it starts to break down the gelatin to where the gelatin is no longer solidified and it is just liquid, then we know that that organism has gelatinase. And if it has gelatinase, then we can narrow it down to perhaps it's in the genera Proteus or Serratia. If it doesn't break that down and it's still very thick gelatin, then we know that it is not one of these, and we can look further in a different direction. Another example is casein. And so casein, right, is in milk, and ACE is an enzyme. So in this case, this enzyme is going to degrade casein in, for example, skim milk agar. So utilizing skim milk agar is a good way to identify particular bacteria or microorganisms that have caseinase. So another way that we can identify. So then once we get to these, so at the point of the extracellular proteases, right? So these extracellular proteases exist on the outside of the cell. They go into these very specific sequences, and they're going to break apart the amino acids at these specific sequences. Then what we have up here are these fragments, or these peptides, that are going to be taken up and brought inside the cell. So after uptake of peptides by the cell, then these protein fragments are deaminated. And deaminated means to remove the amino group. So remember, they're amino acids for a reason. They have an amino group on them, but then they also have different types of side chains, and that gives us the specific type of amino acid. So they are deaminated inside of the cell. And then this amino group is then going to be moved and perhaps reused or recycled. And then the rest of that is going to enter the transition reaction, or the Krebs cycle. So the amino group, we can say, is reused, and then the remaining parts can go into the transition reaction or the Krebs cycle, depending on the side group or the side chain. So that's very specific to the different types of amino acids. So that wraps up our catabolism of lipids and catabolism of proteins, and actually that wraps up our catabolism of all of our micronutrients here. We're not going to utilize, or organisms don't typically utilize, nucleic acids. That's our fourth biological molecule. Don't typically utilize nucleic acids for energy, although in some ways some organisms can do that if necessary. It's not something that's done very often. We go through our carbohydrates first, then we go through our lipids, then we go through our proteins. And so we see organisms able to do this as well. So keep in mind that as we're talking about this, again, the reason that we're talking about all of these different things, and the reason that it's important that you know the steps to these processes, is because of identification of microorganisms. It's important to understand how organisms can break down things for food, for energy sources. It's important to understand how they utilize carbon, where they get carbon from, how they get energy. When they get energy, is it from carbohydrates, lipids, proteins? What kind of enzymes they use to do this? Because all of these things are going to kind of culminate in understanding and identifying different microorganisms. And that's what microbiology has done, at least partially, is identifying different microorganisms. And once we can identify different microorganisms, we can study them, but then also kind of in a backwards fashion, if somebody is sick with something, we can utilize these different understandings, and we can develop tests to determine what they are, so that then we can treat them when it comes along. So it's important that you understand these different processes, so that as we move forward in the semester and we're talking about things like different microbial tests for different microorganisms, when we move into things like antibiotics and ways that we have antibiotic resistance and things like that, it's going to be important that you understand all of these kind of systems ahead of time, so that then you can understand how those work as well.